Hello everyone, my name is Emma Hendrix and today I am going to be showing you my 3D representation of my protein PIEZO1. And I'm going to take this video to represent it towards my mom, who is a special education teacher back at home, so doesn't really have much of a scientific background, nor in college was she a STEM major. So I'm going to go ahead and get started. So again, this protein is PIEZO1, and PIEZO1 is known as a mechanico-sensitive ion channel, which basically means that it's a protein that sits in the membrane of a cell when the cell experiences physical pressure or stretching. And in this body, the protein is located in the cell membrane, which is a thin layer that surrounds and protects, as you can see here, the colored part surrounds and protects every cell. When PIEZO1 channels open, it allows small particles known as ions, especially calcium, to enter the cell. And here I have, as you can see, a green jewel and a pink jewel, and these represent the ions going into the ion channel. The cup is also known as the ion channel, and the green jewel is represented as calcium, and the pink jewel is represented as sodium. And these ions can help the cell communicate and respond to physical forces in their environment. PIEZO1 is especially important in red blood cells and blood vessels because these cells are constantly experiencing mechanical forces that helps blood move throughout the body. And however, when this mutation occurs in the gene that makes PIEZO1, the protein may not function properly, and certain mutations in PIEZO1 are associated with a disease known as dehydrated hereditary stomatocytosis, which is a condition that affects red blood cells. And in this disease, the cells lose too much water and become dehydrated, which can lead to so many problems with how oxygen is transported throughout the body. And so to help visualize the structure of this protein, I used a bunch of household items from my house. And the overall shape of PIEZO1 is very unique and is often described as looking like a three-bladed propeller. As you can see here, we have one propeller, two propeller, three propeller. And this protein is actually made of three identical subunits that come together to form a single channel. In the model, the three objects here, the popsicle stick, the straw, and the furry blanket type thing, represent those three protein subunits, and they curve outward in a way that helps reflect the real shape of the protein. And in the center of the model, I used this plastic cup to represent the ion channel port, which is the opening where ions pass through the membrane when the protein is activated. And the cardboard base that I use here for the little plate is underneath, represents the cell membrane, showing where the protein sits in the cell. And proteins have several levels of structure, and I tried to represent all of these levels to the best of my ability in this model. So the first level we have is the primary structure, and this refers to the sequence that amino acids make up the protein. And amino acids are small building blocks that link together to form a protein chain. And in my model, I use these colored beads down here in a connected line to represent this sequence of amino acids. The second level is the secondary structure, which describes how parts of amino acids can fold into specific shapes known as alpha helixes and beta sheets. These structures form because of the interactions between different parts of the amino acid chain, and in my model, I used a twisted foil pipe cleaner to represent an alpha helix, as well as a beta sheet with this balloon. And the third level is the tertiary structure, which refers to the overall three-dimensional folding of the single protein chain. And each curved object here in my model represents one fully folded piezo protein subunit. Finally, the fourth level is the quaternary structure, which occurs when multiple protein subunits come together and they form one functional protein complex. And PIEZO1 has this level because three separate protein subunits assemble together to form the ion channel here in the middle. And in this model, it's represented by the three objects surrounded by the cup. The mutation assigned to this protein is called V2476N, and this means that a specific position in the protein, one amino acid called valine is replaced by another amino acid called methionine. Even though this might sound like a small change, replacing just one amino acid can sometimes affect how the protein folds or functions or the shape. In my model, I represented the mutation by using different colored beads, as you can see, in the amino acid chain, where I used this black bead or black button instead of a colored button to represent the mutation that comes from this protein and where the amino acid is located. In a normal cell, PIEZO1 only opens when the cell membrane experiences mechanical pressure or force, such as when blood cells move through the blood vessels. However, certain mutations can cause the protein to behave differently, and some PIEZO1 mutations cause the channel to stay open longer than it should. When this happens, too many ions can enter the cell, and this disrupts the balance of the ions and the water in the cell, and in red blood cells, this imbalance can cause the cells to lose water and become dehydrated, and this can just damage the shape and the function and their ability to carry oxygen throughout the body. Although this model doesn't generally show the exact structure of PIEZO1, there are several limitations to this. But first, the real protein is like incredibly small, microscopic, very small, and made of thousands of atoms, while this model is definitely a simplified representation made of everyday materials. Second, this model does not accurately represent the true scale or proportions of this protein compared to the cell membrane. Thirdly, proteins are dynamic structures, meaning they constantly move, change shape, something's always happening with a protein, and my model can't show how PIEZO1 channels actually open and close in response to force or mechanical pressure. And even with these limitations, this model still does a very good job of showing the overall shape of the protein and understanding how these mutations can affect the functions. And that is all that I have for you today. Mom, I hope that this helped, and I hope that I was able to better explain PIEZO1 by providing this 3D model of a representation with all of my structures and the other parts of PIEZO1 along with the mutated mutation gene along the amino acid. Thank you so much.